Hello everyone. So today we are going to learn about morphology of bacterial colonies. Okay. So before moving forward, we need to understand what is a bacterial colony, right? So the colonies, when we hear about the term colony, that means the colonies that are found on the solid media. Okay, so the colonies of bacteria thrive on solid media. They cannot be grown on liquid media. Okay. So, this crucial characteristic means this morphological characteristic of this bacterial colonies, they act as a crucial identifying criteria because it helps to identify the microorganisms. Okay. So, what is colony? It is a visible mass of microorganism that emerges from a single mother cell. Okay. So, when you grow a culture on the plate, so there's a single bacteria. From single, it will go on dividing in the form of 2 to the power n, and they will form a group of microorganisms together, which will be visible mass. Okay, which will be visible and visible mass of microorganism, and that will be called as colony. Okay. So basically, the morphological characteristics that are present that we study are size, form, texture, elevation, coloration, and growth media that define the colony formation. Okay. So if we summarize it, what is the colony morphology? That is, it is the cultural characteristic of a bacteria on an agar plate. Okay. So what is the purpose of studying the colony morphology, right? So these colonies are made up of many microbes. As I said, they are made up of many microbes. They divide into the one bacteria from single bacteria. They divide into two, two to four, two to four to eight, and thereon they go on dividing. Therefore, they share the same genes. So these are the colonies. So one single colony can be said to have been made up of many microbes, similar types of microbes. So, in order to determine the species of a microorganism, researchers replicate the bacteria into a colony. Okay, since a single bacteria cannot be seen through the naked eyes, is not visible to our naked eyes, therefore, that single bacteria is incubated and is allowed to replicate and form colonies so that we can see it through our naked eyes and then we count it. Okay, so this colony morphology can sometimes be useful in bacterial identification. Okay, so this colony helps us in counting the number of colonies that are present on the agar plate, and also it sometimes is also useful for bacterial identification to identify the microorganisms. Okay, so the morphological characteristics that we study in this colony morphology are of 7 types. So the first one is size, second is form, third is texture, elevation, margin, color, and the last one is appearance. Okay, so these are the general morphological characters that we study for the morphology, colony morphology. Now, first is our size of the bacterial colony. Okay, so this is the first characteristics. So, the diameter of a colony that may be measured in millimeter or described in relative terms such as pinpoint, small, medium, and large. Okay, so depending on the size, the smallest size is punctiform, then is small, third is moderate, and this one is large. So the large colonies are very easily visible to our eyes. Small colonies need little bit of hard work, but it is visible, whereas the punctiform are very vaguely or very blurrily visible, and they are very difficult to count. Okay, so the colonies that are larger than about 5 millimeter are likely to be motile organisms. Okay, so greater than 5 millimeter are motile organisms. Now, these punctiform colonies they can be distinguished from the circular colonies by their very small size. There is a difference between this small size colony and punctiform colony. Okay, so how can we different? The punctiform colonies are very small as compared to the small sized colony. Okay, now this punctiform are the dotted or the pinpoint colonies that are grown on the agar plate. When we do the culture on the agar plate, we will sometimes find that there are some dot form of colony. Okay, dotted, which are very difficult to count. dot or pinpoint. So these dotted colonies are called punctiform. These are the smallest form of bacteria or the smallest size of the bacterial colony. Okay. Now coming to the second worm, is our form of bacterial colony. This is our second characteristics. Now this gives the information about the colony shape that appears from above. Okay, so when we look at the agar plate from the above, we will see this type of forms. So this describes the above view of the colony. Okay, so these are the five types. First one is round, second is our irregular, third is our filamentous, fourth is rhizoid, and fifth is punctiform. These are the types of shapes which we can see when we look over from the microscope. Okay, from above the agar plate. Then is our colony elevation. Now this gives information about how much the colony has risen above the agar. Okay, so for this to see, what you need to do, you need to tilt your agar plate and you have to see from a side view to find the elevation of the colony. Therefore, it is also described as the side view of the colony. Now these are of seven types basically. First one is flat, then is raised, then convex, then pulvinate, ambonate, crateriform, and the last one is Drotsman colony. So these are the types of colony elevation that we can see when we look at the agar plate sidewise. Okay, so the most common is the raised and convex are the most common colony elevation forms. Rest can also be seen in some of the bacteria. Next is our margin of the bacterial colony. Okay, now this gives information about the margin of the colony that appears on the agar. When we have an upper view from the agar plate, we can see the margin of the colonies. Now these are of seven types. First one is entire, second is lobate, third is scalloped, fourth is filiform, fifth is undulate, sixth is curled, and seventh is serrate. Okay, so these are the types of margin that we can see of the colonies. Okay, now these colonies that are irregular in shape or have irregular margins are likely to be motile organisms. This is a fact. So suppose a colony is in the shape of lobate or filiform or undulate or serrate. Okay, so these are the irregular margins colony. So here these colonies may be speculated to be motile organisms. Okay, so highly motile organism that swims over the culture media which is visible with our naked eyes only. Okay, for example is Proteus species. These species are highly motile and they swam over the culture media and they have a colony similar to that of any of the four forms, that is lobate, filiform, undulate, and serrate. Scalloped also comes under irregular margin. Okay, so example is Proteus species. Next is our differentiation based on the color. Now this is the color spectrum, and based on different types of pigmentation of the bacterial colony that gives out the color, the microorganisms can also be differentiated. So some bacteria produce pigment when they grow in the medium. Okay, so for example, the green medium produces by Pseudomonas aeruginosa. Buff colored or off-white colored colonies can be seen of Mycobacterium tuberculosis if grown in algae medium, and red color colonies can be seen of Serratia marcescens. Okay, so these are the types of colonies which can be differentiated just by looking over the agar plate. So this is the differentiation based on color. There is another characteristics in color section, that is opacity. Now opacity is of four types basically. So the first one is transparent. The colony may be transparent or clear. Second is opaque, that is not transparent or clear. Third is translucent, almost clear but distorted vision like through a frosted glass. And the fourth one is iridescent, that is it changes color in the reflected lights. So these are the types of opacity that a colony can have. Now for example, streptococcus species. Now this streptococcus species, they have a pinpoint translucent beta hemolytic colonies. If the Streptococcus species is grown in blood agar, we can find a pinpoint. Pinpoint is what? Punctiform, right? In the size section we have studied. So pinpoint translucent, their opacity is translucent. Beta hemolytic colonies can be seen of that of Streptococcus species. Okay, another microorganism is Staphylococci. It gives a opaque, smooth and circular colonies on the agar plate surface. There is also Mycobacterium tuberculosis. It also gives a opaque, smooth and off-white colored colonies in LJ medium. Okay, so these are the different types of opacity that we can also characterize. Next is our appearance of the colony surface. So how the colony will appear. Now the bacterial colonies frequently appear shiny and smooth in appearance from outside. Okay, so most of the colonies are shiny and smooth, whereas others show dull, veined, rough, wrinkled, or glistening type of appearance. For example, Bacillus species give a dry and wrinkled colonies, whereas Pseudomonas tudsiri also gives a similar appearing and wrinkled colonies. Okay, so both of the species give a wrinkled colonies. The Mycobacterium tuberculosis gives a shiny and smooth colony. Okay, so these are the examples of their appearance. Now the next one is our texture of the bacterial colony. Okay, so there are four textures of the bacterial colony that can be seen: dry, smooth, viscid, and now this dry um, is a type of colony that usually what it does when you try to pick it up with the loop, it breaks apart. Okay, now this dry portion it breaks apart, and that is why this colony is very hard to pick up with the loop. Smooth is moist and little bit hard to pick up with the loop because they're smooth. Okay, viscid is that it sticks to the loop and hard to get off from the loop. It doesn't easily get off from the loop when you try to put it on the agar plate. And the last one is mucoid, which is sticky and mucus like. It is very sticky and mucus like. So these are the four types of texture of the bacterial colony that can be seen. Okay, so this is the complete video on our colony morphology. I hope you have liked this video. If you have any queries or suggestions related to this video, you can give the comment in our comment section. And if you want a detailed notes of this video, you can follow our LinkedIn page. The link is in the description. If you have liked this video, do give a thumbs up and share and subscribe to our channel for more such related microbiology updates. And I'll see you in our next video.